I want to welcome everyone to our October Knowledge Portal Network webinar and workshop. So today, I'm going to share my screen for a little bit and do a little intro, and then I'll turn it over to our guest speakers. I'm going to do a little welcome, set up the conversation, and then I'm going to turn it over to our guest speakers. Today, we're going to hear from Krishna Aragam and Adam Butterworth, two colleagues we've had the pleasure of working with in the past year and beyond for on our cardiovascular disease knowledge portal, which is part of our common metabolic disease knowledge portal. And today we're going to hear about insights from the CARDIoGRAMplusC4D Consortium into risk variants and genes for coronary artery disease. And then after that, Maria is going to take us through the demonstration of these results on the portal in real time, so you can see where you can find them and how it's fully integrated with the other results in the portal. We'll save some time for Q&A at the end, and then I'll do a little bit of a review of what's new and coming from the network over the next couple of months, as per usual. But first, a little setup. I don't have to impress upon this crowd how well I think complex traits have done in the human genetic space, with over 100,000 loci identified for complex diseases. And I named a few traits that are near and dear to our hearts at the Common Metabolic Disease Knowledge Portal. And what's wonderful about this is that these results are readily accessible on our portal, but also on other resources for all common disease, including the GWAS catalog and the GWAS atlas. So that's successful, right? But really our goal is, after these loci have been identified, we want to prioritize genes and variants for follow-up. We want to know what they do, what the mechanism of action is, what is the variant, what tissue, and so on and so forth. And this is a massive effort and clearly going to be the subject of a lot of our focus for the next several years in our field. And there are global movements out there, which many of you are participating in, to help do this. There's the International Common Disease Alliance, which is helping to do this at a sort of coalition of the willing across the research domains and space. And then more recently, the National Human Genome Research Institute has announced a set of RFAs around understanding the impact of genomic variation on function. So there's some big efforts going on. Closer to home, our Accelerating Medicines partnership is focused on just this, for common metabolic disease. And our public-private partnerships mission really is to find promising new targets for common metabolic diseases, including liver disease, kidney disease, obesity, cardiovascular disease, type 1 and type 2 diabetes. And so, that's our goal and that's our focus. But what's really lovely is that so many communities are doing this in grassroots efforts, very coalesced efforts around what is already known about effector genes at these loci we've identified for common diseases. So today we're going to hear one effort, but first I want to tell you what's currently on the portal and how we've been working in this space for the last couple of years. So type 2 diabetes is a wonderful community part of, and really beyond the genetics, the community's really worked to say what is known about, the effector genes we know now. So right now on the portal, you can see effector genes for type 2 diabetes from 3 different sources. One is a curated list of effector genes for type 2 diabetes from genetic, regulatory, and perturbational evidence. And the other are 2 bioinformatic predictions. But we put them all together for you and synthesize them in a way for you to allow to see both the results, but also the supporting lines of evidence and get to the underlying data. Well, we're hoping to do just the same for many other complex diseases. And one of the ones we have had the fortune of working with to do that is the group that we worked with to build the cardiovascular disease knowledge portal, which has been a portal we've had in our suite of portals for about 5 years now. And we've had all the genetics data represented for cardiovascular disease, lipids, atrial fib, heart failure, and so on and so forth. But taking those loci and really transferring into effector genes is really the important work. So Krishna and Adam are now going to tell you about their work in that space, and then we're going to show you how we do it on the portal. So I'm going to turn it over to them, and give me a second to stop share, and then we'll turn over to Adam and Krishna. And there's Adam's face, fantastic. Hey, Adam. All right. Again, if you have questions, please pop them in the Q&A or the chat. We'll pull them together, and we'll have time for them at the end. So with that, I will turn it over to Krishna and Adam. Do I need to allow you to share your screen, Krishna? Or are you good? You're muted still. There you are.
Is that shown okay?
Beautifully.
Let me just
Yes.
present and let me know that I'm on the right screen.
You're good to go.
Yep, that's showing well, Krishna. Great. Well, thanks, Noel. Thanks for the introduction, and thanks to the Knowledge Portal team for inviting us to come and tell you a bit about the work that we've been doing and how we've been interacting with the Knowledge Portal team to help make the results accessible to the community. This is a talk really on behalf of the CARDioGRAMplusC4D Consortium, for whom Krishna and I were fortunate to be able to lead the latest gene discovery effort. So we're going to tell you a bit today about what that effort entails, what we found in terms of novel associations, etc., why we think that's important, and particularly then about the approach that we took to try and identify causal variants, causal genes, and causal pathways across all of the signals that we found for coronary artery disease, before we then see a demo of the portal and how the results look in there. So Krishna, why don't you take us on through the next deck. So nothing to disclose in particular here. Here's the outline that I just mentioned. So why don't we get straight into it, and we can talk a bit about the background. As people on this call probably know, but just in case, coronary artery disease, of course, one of the leading cause of mortality worldwide. So still an important condition. We have quite a lot now of effective and treatments that we can use that are widely used, but we still see a huge number of heart attacks. And you know, in the schema on the left there, we see this is relatively we talk about coronary artery disease. Typically here we're thinking about heart attacks, myocardial infarction from a blocked artery, plaque rupture, causing some kind of damage to the heart muscle here, and in some cases even death. So it's still an important condition for us to study and an important one to represent in the common diseases, this catalogue. So on the next slide, you can see a little bit about the history and the timeline, the kind of context of where we're placing ourselves in the history of genetic discovery for coronary artery disease, from 2,000 or 3,000 cases back in 2007, finding the first genome-wide loci, ever bigger sample sizes, several different consortia, so both the CARDioGRAM and C4D Consortia publishing in 2011, and coming together under this this framework of CARDioGRAMplusC4D, slightly mouthy title. And you can see that by 2015, we're talking about 60,000 or so cases amassed from many different studies around the world. More recently, there's been efforts through different kinds of arrays like exome arrays, metabochip arrays, and kind of adding on different individual studies like the UK Biobank study. And so really the goal of our project was to see, could we take the pre-existing data from CARDioGRAMplusC4D and could we add more of these kind of biobanks, more different studies? So going beyond just the case control studies that typically made up CARDioGRAMplusC4D, could we harness things like hospital biobanks, population biobanks, clinical trials, any other source basically that might have a large number of cases of coronary disease and controls that were all genotyped. So on the next slide, you can see a bit about the design of our study. Well, in 2015, we took the CARDioGRAMplusC4D data from then, which had 60,000 cases, and found 58 independent variants reaching genome-wide significance. And then we said, what happens if we add a ton more cases? So we added a whole set of different types, as I mentioned. So we added the UK Biobank and other population biobanks like Decode Genetics, Biobank Japan. We added several clinical trials from the TIMI stable that have identified either high-risk participants or patients who already had coronary disease, and have then since genotyped them, and we got permission to use those and to try and carve out case non-case comparisons that we could informatively use. We had hospital biobank, the partners biobank there that's been recruiting patients coming in through the hospital system, phenotyping and genotyping them, and several more case control cohorts that we could use that you see in the top right there. So basically studies that didn't already overlap that might give us some useful information to distinguish between variant frequencies and cases and controls. On the next slide, then you see the results of that. So to the 60,000 cases that were already published, that we had the summary stats for, we were able to add about another 120,000 cases of predominantly European ancestry that you see in the blue box there, as well as published data from the Biobank Japan study, which added about another 30,000 East Asian ancestry. In total, we had about 200,000, well, more than 200,000 cases of coronary disease and about 1 million controls. And so we put that together in a genome-wide meta-analysis, and now found about 280 independent genetic variants reaching genome-wide significance. And so Krishna will tell you a bit more about our initial findings.
Thanks, Adam. So as Adam mentioned, we're fortunate to be able to study a healthy number, well, a large number of cases here of coronary artery disease, and a substantial bump in the number of genome-wide significant signals that we were able to uncover. And so in total, it was 279 independent common genetic variants that were found to be associated with coronary artery disease. And this translated to 68 novel regions of the genome associated with CAD. And a sizable portion that fell within the 1% FDR range, about 900 or so associations. So again, a substantial boost from at least certainly the last large international collaborative using 1,000 genomes data a few years ago. And a few of the, I guess, high-level insights gained from these analyses, I think that one of the main takeaways was that with this increase in sample size, we see a substantial increase in the number of signals uncovered. And in fact, that seems to scale reasonably when we trend back over time. This is, as Adam showed initially, in that timeline, we date back to about 2007, the first set of genome-wide association studies for coronary artery disease. And we've had ever-expanding numbers of cases of coronary disease studied, and we've had an ever-expanding number of genetic association signals. And now as we add not just European ancestry populations, but trans-ethnic cohorts, it seems that the number of associations will continue to increase. And we certainly saw that in our study, even compared to the past few years. There's been a substantial increase in the numbers of variants uncovered, and it seems like no reason to expect that that's going to go down substantially. Probably we'll just see more and more as more of these cohorts coalesce. But another key insight gained from this study is that while the numbers of associations steadily rises, the number of those that are of most of those associations confer fairly modest risk of CAD individually. So fairly modest effect estimates here. We're saying at least odds ratios less than 1.05 for each of these SNPs. And so what you'll see here is a plot characterizing the relative effect sizes of the various associations that we uncovered, over 200 or so. And in blue, you have what was previously reported as genome-wide significant, and you'll see that some of the outlier or slightly larger effect associations were the ones that were reported previously, probably initially 1.2, 1.3, et cetera. But the novel associations in red, and even the 1% FDR ones, which are probably the ones that will rise above the surface in forthcoming iterations, all tend to be with odds ratios in the 1 to 1.05 range. So suggesting that we'll probably have more and more loci and association signals uncovered, and most of them will likely be in this modest to weak effect range. But it's, I think, important to recognize that just because an association has a modest effect doesn't mean that it's not going to be useful in terms of understanding not only CAD biology, but potentially making inroads into therapeutics. And one good example of this was that in this study, we uncovered the HMGCR locus uncovered. Really, we found that a signal at the HMGCR locus achieved genome-wide significance for CAD, but with a fairly modest effect estimate, again, also within that sub 1.05 odds ratio range. But this is of high importance because HMGCR encodes HMG coreductase, which is the target of statin therapy. And as most of us know, statins are probably the most widely prescribed drug for CAD, for the prevention of primary CAD, but also the prevention of recurrent CAD. So whether you're trying to prevent CAD from occurring or treating those who've already had a heart attack to ensure that they don't have a recurrence and worsening of their condition, statins are given. And so it's encouraging in some ways a validation of this GWAS approach that we now uncover this signal. But at the same time, it tells us that we do have to dig. This was not the first signal that emerged for CAD. Clearly, this is probably the 200 and something signal that's emerged for CAD at genome-wide significance. So it tells us that there is certainly gold if we go look for it, but it's important to ultimately understand the mechanism behind these signals, not just look at their effect estimates. So in thinking about mechanisms one approach that we commonly use now is to conduct a phenome-wide association scan. And so we conducted this in the UK Biobank for all of our genome-wide significant associations. And there are a lot of different results that one could pick at here, but thematically, one of importance is thinking through whether the signals tend to fall under the rubric of traditional cardiovascular risk factors or other non-traditional cardiovascular risk factors. And I think that's something that we tend to look at just to try to classify whether or not there is whether the biology behind a particular variant or locus is one that we might predict or already know, or it's something that's really quite new and representing a new mechanism. And what we found is that the majority of our associations did not associate with known and standard cardiovascular or CAD risk factors like lipids, blood pressure, type 2 diabetes, or adiposity, suggesting that there are probably these non-traditional mechanisms that mediate quite a bit of CAD risk. And so that, again, just gets to the main point, which is that for a lot of the genome-wide significant signals, and this is the full list of all the different loci that have been uncovered prior to our study, we try to bucket them into different mechanistic pathways, but most of these associations are pretty unknown. And so I think really the rate limiting step here is the functional characterization. And usually that starts with understanding, mapping that region of the genome to a specific variant, and then a gene and a mechanism to link it to coronary disease, and understanding how to mitigate then that risk. So that ends up being the real goal of this study, or was the goal of this study, and why we're talking about all of this here today, which is that after conducting a pretty robust meta-analysis of coronary disease with 200,000 or so cases, a lot of different post-GWAS analyses performed, but we focused on a couple, which is, can we do a little bit better job with some of the techniques on hand and some newer techniques to identify the specific variant that's mediating the risk conferred for CAD, and can we do a bit better job of identifying the particular gene or genes that are mediating that risk. And so that's what we will talk about now. And so we'll start, and Adam will start by talking about our approaches for causal variant analyses.
Thanks, Krishna. So as you say, important to try and understand which might be the causal variants, the causal genes, and the causal pathways to disease. We started with the variants using the FGWAS, or functional GWAS approach that Joe Pickrell developed. If you go on to the next slide, Krishna, that tells you a little bit more about it. And the idea here is that one can, rather than treat every SNP independently or just on the basis of its statistical means, one can look at to what extent can we identify tissues or cell types in which chromatin-derived states appear to be enriched for signal. And one can then use that information to try and upweight variants that lie in those annotated regions. So here we took data from the Roadmap Consortium, and more as kind of proof of principle, having seen this before in previous studies, we just showed that if you look at various different tissues and cell types that are available there, you could find about a twofold enrichment for the signals we see in our data in 10 different cells and tissues, many of which have been shown before to be play some role in etiology of coronary disease, such as aorta or liver, adipose, etc. And we can then look, you can then divide the genome up into regions and say which of these regions seem to have significant enrichment across the different cell types and tissues. And you see on the right here the distribution of those. So most of the regions that we looked at seem to have enrichment only in a small number of tissues, but there are some that appear to be significantly that appear to co-localize, if you like, with these epigenomic regions across many of the tissues, and in 3 cases, all 10. So having established that there's some sort of enrichment here for these regions, and we think that these cell types and tissues might be useful for prioritizing which variants are most likely to be causal, we can then apply the FGWAS approach using this information and try to statistically fine map each of the regions to get posterior probabilities and to work out which variants, how many variants would we need to come into a 95% credible set. What's the posterior probability for each individual variant, and therefore are there some that have really high likelihood of being causal. And so the quantitative results are summarized really on this slide. So there are about just over 100 regions for which we could see enrichment. So this doesn't work for all the regions, but for the ones where there is enrichment, we can then try and find that. And we found about a third of those, so 42 of those, had quite a small number of variants in the credible set, so fewer than 5. That was an arbitrary number, but based on discussion with some of the experimental scientists, we said, what sort of number would you be happy about if you were taking these forwards into the lab? So we think for a reasonable proportion of them, we can actually do pretty well. And even in terms of finding variants that have individually very high posterior probabilities. So nearly 50 regions had a single variant with a very high posterior, greater than 0.5. So a pretty high likelihood that that might be the causal variant here. If you go on to the next slide, I think you can see some examples of these regions. So this plot tries to display the 49 regions that each had a high posterior probability signal, and it shows you as well the number of regions that the number of tissues or cell types that that region was associated with, and those are the colored lines that join on the circular plot. As well as around the edge, you can see the numbers of variants that were in the 95% credible set, and you can see that for some of them, there's literally just a single variant that was fine mapped into the credible set. And many of these were well-known variants that we've seen before, such as the R46L variant for PCSK9, for example, or other missense or coding variants that have been well characterized before in the literature. So this gets us some way to understanding what's going on at the regions, because this can help us pinpoint which are the most likely causal variants. But of course, that on its own, it might tell us something about the tissue and cell type as well, because of the chromatin enrichment here. But it doesn't tell us yet which is the causal gene or indeed the causal mechanism. So that was the next stage of our analysis to try and work those out. And Krishna is going to tell you a bit about one of the methods we used for identifying causal genes.
Thanks, Adam. So as Adam mentioned, going from causal variant then now to causal gene, which ended up being the bulk of this analysis, we employed a number of different approaches, but one in particular that's more novel and that might be familiar to several in this crowd and that we've spoken about in the past, but just to review the polygenic priority score, or POPS, which is arisen from Hilary Finucane's team here at the Broad, and just some of the summary or background slides, courtesy of Hilary as well, which is that the main concept with POPS being that it provides an orthogonal way to help prioritize particular genes of interest for a given disease, given that there are a lot of different ways already in existence to help prioritize genes at a particular locus that comes up as genome-wide significant. But this slide really hammering home the point that pretty much all of those methods rely on proximity and rely on local signals. So of course, the simplistic but actually quite effective method is whether the gene is, how close the gene is to the sentinel variant, so nearest gene. But then even other methods like looking at fine map coding variants and EQTLs, local functional genomics, and even some of the existing similarity-based methods tend to rely on distance and looking at what's generally around that given region. And what is generally missing then from that framework is thinking through polygenic gene set enrichments. And that's what the POPS method looks or now currently looks to leverage. And so the basic schema of POPS, which again is a gene prioritization method that leverages the full genome-wide signal and to help nominate these causal genes. It leverages the polygenic enrichments of a number of different gene features, which are illustrated or encapsulated by this matrix here and listed. Some gene expression data on curated biological pathways, PPI protein-protein interaction network data. All of this is used to compute a polygenic priority score or POP score for each gene, which essentially then says that score is, again, specific to the phenotype of interest, in this case, coronary artery disease. And so that's indicating, based on the totality of the information across the genome, how likely that gene is to be an effector for that particular phenotype or coronary artery disease. And in, one of the POPs, which is out as a preprint now, in that iteration, a key piece of it is that this prioritization is independent of GWAS data at the locus itself. There's a leave one chromosome out feature to try and look at the rest of the genome, not the region or even the chromosome where the main signal lies. And so this is, again, really trying to emphasize that POPs is providing information that can and should be complementary to the existing approaches that we use. And so how do we then take this forward? We have again, we take GWAS summary statistics and combine them with this gene feature matrix specific to the phenotype, and create POP scores for all the different genes that are passing, especially a certain threshold. And then now that all those genes have been scored across the genome, we'll then intersect this list of scores with our GWAS hits. And so we'll say, now at this region, or at around this sentinel, which gene has the highest POP score? And for sure, we need to establish a window there, so there is some sense of looking at within a particular region, but within that, still, what the prioritization of those genes within a region are now based on genome-wide data. So that's the principle. And so, this being the case, we've had a chance, been in a fortunate position to essentially beta test POPs since its inception in a very focused manner for CAD. And what we were fortunate to see early on was that POPs did prioritize many established CAD genes, and genes that are of relevance to CAD for different mechanistic reasons, whether they be lipid-related genes, genes that are involved in vascular function or integrity, or those that have to do with inflammatory processes. So that was very encouraging to see. But not only were these prominent genes prioritized by POPs, but they seem to be prioritized for the right reasons. And so, as I mentioned, there are a lot of different inputs that go into POPs, and one aspect of POPs is that all the different features that have nominated a given gene for its relevance to CAD are then part of the output that one can review. And so what we'll see here is that for genes like LDLR and ApoC3, not only were they prioritized, but they were prioritized by POPs for, quote unquote, maybe the right reasons. LDLR being prioritized for lipid, or LDL specifically, reasons, and ApoC3 more so for triglyceride pathways. And that all makes good sense and is what we know, so good validation. And even non-lipid mechanisms, GUC1A3 being nominated by POPs for what appears to be, when we take a look at these features in aggregate, probably nitric oxide mediated pathways, which is what we believe to be the case. So again, good validation that POPs is on the right track and not and providing information that could potentially be complementary. So that's looking at individual genes, but what about looking at the totality of the data and what it can tell us about pathways, since that's also of importance to us. And so a clustering analysis in POPs is another feature or another basically another aspect of POPs that helps us to nominate potential pathways of relevance to CAD. And this is, again, a clustering algorithm that produced 3,000 clusters based on the approximately 21,000 features that were input into the model. And then those clusters were again ranked based on how on the contribution of the different aspects of those clusters to the POP scores for the prioritized genes for CAD. And again, it's encouraging to see that the highest ranking cluster was having to do with homeostatic regulation of blood lipids and other top clusters that are known to be relevant for CAD, proliferation of endothelial and smooth muscle cells, and a number of different metabolic pathways. But then you also see a few interesting pathways that maybe are a bit more novel, where you see clusters highlighting early developmental processes and particular signaling pathways like nuclear signaling that involve the cell cycle, which are not as well established, but given these results and some of the reports that we've seen suggest that they might be pretty important mediators of CAD risk. So another thing that POPs is able to provide us with regard to, again, thinking about not just the gene, but the pathway that might be of relevance. And we'll get back to a specific example where kind of this all comes together. But as I mentioned, POPs is one method, and there are many others, but how can it be useful alongside the others is the key. And here's one example where we looked at a recently identified, this is not novel by our study, this is one picked up, a signal identified as genome-wide significant for CAD maybe 4 or 5 years ago, but where the causal gene was not clear. And so this is a signal at chromosome 15 where you can see the peak is sitting right in between a few different genes. And so you could turn to a lot of our typical methods to try and prioritize a causal gene here, but the gene expression data points a few different ways with cis-QTLs in GTEx and StarNet for ABHD2, MFGE8, and this HAPLN3. So when you turn to our known methods, you kind of get a bit of a tie, and so it's a bit of a toss-up as to which one is the most likely causal gene here. Whereas POPs identify this MFGE8 or nominated MFGE8 as the top-ranked gene, and we have since seen that there's actually some fairly recent functional work whereby CRISPR-Cas9 in vitro deletion of the region modulated expression of MFGE8, but did not change expression of those two other genes. And when you knock down MFGE8, it resulted in reduced smooth muscle cell monocyte proliferation in the coronary. So seemingly some pretty good validation of this for this particular gene as being a likely effector of CAD risk, and potentially a good example of how POPs could be used to break a tie, or at least provide complementary information alongside what we use to date to nominate causal genes. So with that, basically we've got POPs and then we've got several others. So the whole point here being, I spent a bit of time on POPs just because it's new, but really it's one of many methods, and it could be helpful to break a tie, but really have to consider it as part of the lineup. And so what was that lineup? And that's what I want to address here for us. How do we go about this systematic causal gene prioritization using POPs and several other tools? And the long and short of it, I guess we can say that Adam and I have thought about this quite a bit, and we've tried to slice it in many different ways. And the answer is you really can slice it in a lot of different ways. And you can have a lot of different inputs into this. You can also use those inputs and weight them differently or have them unweighted. So tons of different combinations. But we'll tell you what we did and how that looks. And I think it'll be good to have a discussion afterwards about those different possibilities. I think that's really one of the main goals of this session, is that ultimately we're capturing all this on a portal so everyone can peruse it and give us their take and maybe modify for the future. But so what did we use? So we had 8 predictive features to help prioritize likely causal genes for coronary artery disease. And I've listed all 8 of them here, and I'll go through all 8 of them, but I think it might help to thematically to categorize them in a particular way, which is to say that for a couple of them, in the top left, you've got nearest gene to the sentinel variant. So that makes good sense. That definitely needs to be one of the 8, but also point to the one on the toward the bottom left, where you see gene has a protein altering variant. So just means that there's a protein altering variant in high LD with the sentinel variant. I flagged these 2 because they're fairly disease agnostic, so to speak, in that they're looking at location and then what the properties of that variant in particular. Whereas the remaining 6, which I've starred now, are a bit more disease specific. And so that might be one way to thematically categorize these predictors, and really the fact that the majority of what we're looking at here is more disease specific. And we found that to be of benefit. So what are these? So we looked in particular to see if there's a gene region causing a relevant monogenic disorder. And what do we mean by relevant monogenic disorder? It's phenotypes related to coronary cardiovascular disease, CAD, blood lipids, blood pressure, bleeding disorders, or other vascular abnormalities. We looked to then Mendelian randomization studies of gene expression, protein levels, or other proximal phenotypes that implicate specific genes as causal effectors for CAD or cardiovascular diseases, and also relating to cardiovascular drugs targeting proteins encoded by that gene. So again, very disease specific. We've talked about POPs and the fact that the POP scores that we're using to guide us are tailored to CAD. So that's certainly very disease specific. And then looking at EQTL data, in the StarNet in StarNet and GTEx, StarNet being a study of 600 coronary artery disease patients, so fairly specific there. And the tissue types that we looked at, atherosclerotic aortic root, the intramammary artery, blood, liver, subcutaneous fat, and abdominal fat, all these are highly relevant to coronary artery disease. We also looked at relevant mouse knockout phenotype data and looking at, again, phenotypes for cardiovascular physiology and morphology, as well as related to risk factors such as lipids and body weight. And then finally, we looked at genes with a previous rare variant association, specifically with coronary disease, either individually or with aggregate gene-based testing. So these are the 8 features that we used, and again, purposely kind of categorizing them as to maybe a couple that were disease agnostic and the majority being a bit more disease specific. And so again, there are different ways to then combine this data. How do you combine it? Do you weight something differently because for some reason, and there are probably endless possibilities. We chose to go the simpler route and say, we've got a number of these features, let's see how they align, and let's take the unweighted sum of all these predictors and look to see where the evidence converges and use that to help guide us. And so we ultimately took the unweighted sum of these predictors and based on that said, based on we were able to prioritize at least one likely causal gene at 206 of our associations based on having at least 2 concordant predictors. And because a given more than one variant could be associated with one gene, it ended up prioritizing 185 distinct genes. And so what you see here in the middle is a plot for the number of concordant predictors that went into prioritization of a given gene. So for 92 of those genes, they had 2 lines of evidence pointing to the same gene, all the way up to 2 genes where there were 7 concordant lines of evidence. And so you can see that a little bit better in this heat map and list here, where you've got the top genes based on number of supporting features, and again, encouraging that some very known players in CAD risk like PCSK9 and LDLR and ApoB, LPL, IL-6, et cetera, are all making this list here with lots of supportive features lining up to nominate them. And so what are some other just key summary metrics of this before we go into examples? It's good to know, it's good to see what the yield was on of this approach for prioritizing positive control genes for CAD, which we picked based on consensus across our large international group. And out of 30 positive control genes, this approach correctly prioritized 28 of them. What are a couple other metrics to look at? So now you'll see in the bubbles here that I put in a bunch of numbers, and this is really looking at the agreement essentially of that particular piece of information with all the other pieces of information around a given sentinel. So the denominators are different because you've got information from nearest gene for all the different sentinels, 185 total, and same for POPs. But you may have relevant information about a monogenic disorder or EQTL data or mouse knockout data for only select subsets of the variants. So that's the denominator. And then the numerator is the number of times what was called by, say, given EQTL data lined up with the consensus from all others. And so calling that agreement, we saw that agreement ranged from anywhere from 66 to 84% across the various predictors. Another metric that was of interest was just to say, well, what about between any two predictors themselves? How often do they concord? And so really the concordance there between any two predictors range from 29% to 73%, which we thought was interesting, the 29% in particular, suggesting that there probably are pretty complementary lines of evidence here. It's not that these are all providing the same types of information. There can often be competing information. And so, it really does, at least in our framework, lend itself to a majority rules type construct. And at least that's what we went with here. So those are some of the basic stats of our approach. I think it'll be good to have some discussion, but before we do that, Adam and I want to go through a couple of exemplar loci to try to bring some of this together, and that might then help guide even some of the demonstration.
Thanks, Krishna. Yeah, we'll just round off with a couple of specifics, having presented the sort of overall. So the first one is a gene called ITGA1, or as you see here on chromosome 5. And this one was sort of strongly prioritized by the fact that our lead coronary artery disease SNP that you see here circled in black is also a lead SNP for EQTLs for ITGA1, but no other genes in relevant tissues such as liver. You see here also weaker for aorta, fat, artery, etc. So that gives a good starting point to point towards ITGA1. If you go on to the next slide, Krishna, you see that the lead SNP here, and I should say the lead SNP here had a very high posterior probability of being the causal variant. There were two variants that rose out from the noise of the other variants tested in the region, but this one lies in these epigenomic marks for several of the tissues, including liver, aorta, fat, etc., whereas the other one only lies in those for a couple of tissues. So this one I think had a posterior probability greater than 0.9 of being the true causal variant. And so you see that the CAD risk allele here decreases ITGA1 expression, notably in the liver, but also in these other tissues. But then when we did a phenome-wide association scan in UK Biobank to see what other downstream risk factors the variants associated with, you can see it has increases in liver enzymes, perhaps consistent with the effects in the liver. It also has weak effects on increasing proatherogenic lipids like LDL cholesterol and total cholesterol, so perhaps explaining what might be the mechanistic pathway, as well as effects on inflammation markers like CRP and albumin. So we think at least for some of these loci, this was a novel one that hadn't been found with coronary artery disease before. We can start to pinpoint the causal variants, the likely causal gene, and perhaps derive some hypotheses that can then be tested about the downstream pathway to disease. If you go on to the next slide, Krishna, then you'll see that it's maybe not a surprise that ITGA1 comes up, integrin alpha-1, because not only does its binding partner also come up amongst some of our loci, but there are several other integrins that are all closely related in this interaction network that you see here, which all had signals either at genome-wide significance or as part of the 1% FDR set that Krishna mentioned earlier on. So, of course, these loci don't act in isolation, but when we look at them in aggregate, we might pinpoint specific pathways or networks that are involved. So integrins sit on the cell's surface and are responsible for attaching to things like laminins and collagens, and so there's plausible biology here. We think here, rather than just writing a paragraph in the discussion about the biological plausibility, we can go a little bit further by trying to use the systematic and sort of objective approach that Krishna's outlined to try and pinpoint which is most likely to be the causal gene, and then to try and interpret the biology afterwards. And Krishna is going to tell you about the second example that we've followed up in some detail.
Thanks, Adam. So I think the ITGA1 example is a fantastic one of all these different lines of evidence converging that we looked at. Another one that we saw that was of interest was this signal on chromosome 19, this RS7246865 sentinel is very gene-dense region of chromosome 19, about 13 different genes within 500 kb here. The nearest gene is this myo9b, but there are many others in the vicinity. There's GTEx, and actually myo9b encodes this myosin protein with Rho GTPase signaling activity, so it's involved in cell migration. We end up having cis-EQTL data to also support myo9b. Notably, there is, I think what I didn't mention here is that we also had a high PPA variant that was that we the lead SNP here had a very high PPA, and so that helped us in terms of prioritizing, what might be what we could interrogate or perturb here. And so between the POPs here actually prioritize myo9b as the top ranked gene above all others. And what was interesting was that the features that prioritize POPs had to do quite a bit with cell motility, cell migration, essentially saying that this region seems to be of relevance for CAD because of cell motility, cell migration, particularly with vascular cell types. And because of that, it prioritized myo9b as being consistent with that phenomenon. So given that now we had maybe a likely causal variant with a high PPA and some multiple lines of evidence converging on a potential likely causal gene and pathway, we actually took it one step further with some of our colleagues and pursued more functional interrogation of this region of the genome, where what we found is that this particular CAD signal happens to be in a non-coding enhancer region when we looked at all the relevant tissue data. I'll just summarize this very briefly, which is to say that for the sake of time, which is that when we actually look at that particular enhancer and delete that enhancer by CRISPR-Cas9, what we see is that there are, we looked at all the genes in the territory, and there were only 2 genes where in CD relevant cell types, endothelial cells and vascular smooth muscle cells in particular, we saw changes in expression. We saw for ECs changes in myo9b and house8 expression, and for vascular smooth muscle cells, a change in myo9b expression in particular. No major change in expression of any other genes in the region, no change in any genes in monocytes. And so then we took it a step further then and said, well, what is this enhancer, which seems to be having some effect on these couple genes here? What might be the pathway of relevance mechanistically to CAD, and what might be the cellular phenotype? And so we know that there are some cytoskeletal functions of myo9b and house8 in other cell types. So we looked into the effects of these genes in a wound healing, a monolayar wound healing assay, which looks to encapsulate both cell migration and proliferation. And what we saw is that, in particular, enhancer deletion in ECs caused impaired wound healing, and as did knockout of either myo9b or house8. So this is now suggesting and maybe even putting together that at this region of the genome, where there is a human genetic evidence to support association with CAD, we are seeing that perturbation of an enhancer seems to affect 2 genes, both of which might be implicated in migration and proliferation of CAD relevant cell types. So now getting that much closer to linking this region of the genome to the downstream biology and CAD risk. So with that, and just to make sure we have time for the rest of it, just to summarize that, I think the three main points that we want to communicate here from this study was that certainly with larger and larger sample sizes, we will most certainly identify many more CAD risk variants with likely weak individual effects, but still could be quite informative in terms of pathways and potential therapies. I think we've learned that there are probably lots of different ways to do this, but whichever way we pursue, we need to have a very rigorous approach utilizing lots of different inputs and preferably disease-specific inputs to help prioritize causal variants, genes, and pathways. And that as much as we, and I think that having all this data generated, having it housed on the portal, as is going to be demonstrated, is the key, is a real key. And the goal is to create these testable hypotheses for experimental follow-up. And we like to show that, I think that we have at least one example of where we had lots of interesting information about a region of the genome, but thirdly and critically, we still needed the experimental interrogation to really best understand the mechanism that links a signal to coronary disease risk and the specific mediators of effect, because without it, we may not have uncovered house 8 there as a potential player. I think we thought it was quite a success that we were able to guess, quote unquote, that myo9B seems to be implicated and even predict potential pathway. But without the experimental interrogation, we wouldn't have had a complete picture to follow up on further. So it's with that again, we'll just thank all the different folks that have been involved in this study outside of Adam and I, which are many listed here. In particular, a lot of collaborators here at the Broad and the University of Cambridge who have really helped to co-lead a lot of this work. So thanks all, and looking forward to the demonstration.
Great, thanks, Krishna and Adam. Let me share my screen.
Great, we've been answering questions as we go in the Q&A, so that's been good. So we'll let Maria take the line to the time to actually demo these results. That was a great setup, Krishna, for the portal in terms of what we're hoping to do.
Okay, well, this will be really brief, but let's just take a look at what you can see, some of the things you can see in the portal for this, for one of the examples, ITGA1. We'll type it in. So I'm on the Common Metabolic Diseases Knowledge Portal right now. The Cardiovascular Disease Knowledge Portal is a subset of this, and you can go there to get a sort of more focused view with just the phenotypes that are more directly relevant to cardiovascular disease, but I'm just using this one for now. So when we type in a gene name, we go to the region page. I'm not going to go through the whole thing, but let's scroll down to here to the locus-zoom. Now, when you first go to a region page, the locus-zoom display is set for the phenotype for which there is the most significant association in the region. So, but you can change that. So I'm going to remove LDL cholesterol and put in coronary artery disease. So we can look at that. And I want to stress that we do not yet have in the portal the results of the study that Krishna and Adam have just talked about. We're looking forward to incorporating those. And so that will be a lot more to explore. So right now we just have various other studies, all the previous studies of coronary artery disease. But here, if we look at ITGA1, actually this variant here, which is the top variant for CAD, is that variant that they found in their study, and it's the one that has a PPA of 0.95. So that's pretty good. Using this whole interface, which we're calling the genomic region miner, we can add some annotations and take a look at the epigenomic annotations in the region. And these come from our sister portal, sister database, the Common Metabolic Diseases Genome Atlas. And they're displayed here. The annotations are enriched. We run the Gregor algorithm to determine enrichment of genetic associations within these annotated regions. And we can filter by fold enrichment to look at just the more significant ones, and that narrows it down a little bit. So I want to stress that we're working actively on this interface and we're trying to make it a lot more useful. But just from a really quick look here, if we put the crosshair over the variant, go down here, you can see that there are enhancers annotated in many different tissues in this region, which is also what Adam and Krishna found. Now, if you look on the variant page, so here's this table lists the variants in the region, and this is the one we've been looking at. So let's look at its variant page. You can also recapitulate many of the things that they saw. So for example, PheWAS. This is a PheWAS just across the data sets in the portal, not all of the data sets in UK Bio, not all of the data points in UK Biobank, all the phenotypes. But still, we see a lot of the associations that they observed, such as the liver enzymes and inflammation with C-reactive protein. So that all makes sense. And also at the bottom of this page, we show annotated regions that are overlapping this variant. And again, as they found, you can see that it's in enhancers in all of these different tissues. So real quickly, because we're running out of time, I want to go to the effector gene list. So if we, under the tools menu, predicted effector genes, this is our landing page where we just list all of the different methods and phenotypes for which we have effector gene lists. And so if you click on this one, you get to coronary artery disease. And also want to point out that if you click this view research method tab, there's a quick reference so you can look up and understand better what all the fields mean. But here there's just a graphic that shows the various loci. So the table is organized by locus, and then you see the sentinel variant and a lot of all the other prioritization information that Krishna talked about. And I just want to show you ITGA1 here. You can look at one particular gene by filtering this and just find it quickly. And so as Krishna showed us, it's a novel association, and this was prioritized by POPs. 3 features prioritized it. And you can expand this to show even more evidence. You can see whether or not it had all of these evidence types. And okay, this gene doesn't have it, but if a gene has a mouse phenotype, you'll be able to click on a link there and go straight to the mouse genome database to see the detailed information about that mouse phenotype. So we are almost out of time, so I'm going to stop there and hand it back to Noel.
All right, thank you, Maria. That was wonderful, Krishna, and wonderful, Adam. And we look forward to we're talking with Krishna and Adam offline, because we do want to have the supporting genetic results integrated in the portal, and that's soon to be. They're updating the results as they work towards publishing the manuscript for these data. But the effector gene results are on there, as you saw today. And then I'm going to let us wrap up on time, but I thought the questions have been answered in the Q&A. So if there's any questions that people are burning to ask, I'll give a second to ask them now. If not, I have one more point I want to raise. All right, so hands up on our Q&As. But one nice item is what's really important to us is if you know anyone in the community, or you yourself have effector genes for any complex disease that you want to represent in the portal, we have a very easy and fast way of doing that. In fact, we are about to release our type 1 diabetes effector gene list soon, curated by Kyle Galton and colleagues and represented on the T1DKP. In the same framework that you saw for coronary artery disease and type 2 diabetes, these are curated lists represented from the community that pull in the information from the portal. What's nice about this is we started with a T1DKP with the goal in mind to build a T1D effector gene. So the portal came first, and then a lot of the evidence sources in the portal for the type 1 diabetes portal are also in the effector genes. So it's a lot more incorporation there, which moves the needle again forward to try and represent both the results and the integrated underlying information. So those will be available in the coming weeks on the portal, and look for that. We're also going to be doing this for osteoarthritis on our musculoskeletal disease knowledge portal. Again, same idea. We want to work with the community to understand the science and the representation, just like we did with Krishna and Adam, to represent the results in the portal, but make them accessible in the way that the community envisioned, and also help move that entire needle for all those GLAS loci. So I'm going to break now. I'm respectful of all your time. I'm so glad you joined us. Our next webinar is going to be December 9th. And on that webinar, we're going to turn directions towards our research portal perspective, which is about not just human genetics data, but about representation of other results, including image data and proteomics data and things like that universe that allow us to move towards representing different types of omics data in the future. And one of the central portals in that effort has been this lipid droplet portal that we've been working on to basically represent that well-studied organelle by some colleagues of ours and make that accessible. So that's another frame that will be very valuable in the coming years for the portal. So hopefully you can join us for that. And these will be available on the knowledge portal if you missed this. And I thank you for your time. And I thank Adam and I thank Krishna for being great colleagues and also doing such wonderful work. So thank you, everybody. Take care.
Thanks. Bye-bye.
Thanks, everyone.
Bye.